All right, everyone, we are going to cover the lymphatic system today, chapter 16. Here are some roots prefixes that you will need to know. Auto is going to mean self. Um, we should know that one already from like autoimmune diseases. Immunity, we know that word. Just immune means free or exempt, like resistant to. Inflammatory, the inflammatory process. Inflame means like to set on fire. Patho, like a pathogen, is a disease-causing agent. So a pathogen would be anything that causes a disease. Okay, so the lymphatic system. Basically, it's just a network of vessels that kind of help out the cardiovascular system. Um, it's very closely related to the cardiovascular system. It's almost like a second cardiovascular system. It's like, think of it as like a plumbing system of the cardiovascular system. So the cardiovascular system kind of pumps the blood and creates this interstitial fluid. Remember that 20 liters that comes out and the 17 liters that comes in and the 3 liters remains. We'll talk a little bit more about that later on. And that 3 liters gets picked up by the lymphatic system from the interstitial fluid. And the lymphatic system kind of mirrors the cardiovascular system in its kind of circulation. But all it is it's just a collection of cells that travel around in these things called lymphatic vessels. When we look at the lymphatic vessels, kind of structurally or anatomically, they are going to resemble veins the most, only because that they have valves. We'll get into that a little bit later on. All right, so what does the lymphatic system do? There's really 3 things that the lymphatic system does. And we'll do 1, 2, and 3. So the first thing that it does is going to move and return that interstitial fluid from the interstitium, or that extracellular fluid, away from the interstitial space and transport it back into the bloodstream. That's that extra 3 liters of fluid. The second thing that it will do is transport the production of lipid digestion from the intestine to the liver. So remember that, lipids are fats, and fats and water don't mix. Remember the blood, when we break it down, there's the 45% of the red blood cells, white blood cells, and platelets, and 55% of it is plasma, and of the 55%, 92% is water. So basically, blood is a lot of water, so fat and water is not going to mix, and so we have to package our fats in order to make them be able to be transported in the blood. So the lymphatic system is going to take these lipids from the digestive system back to the liver so that they get transported through the bloodstream. And we do this through the lymphatic capillaries. And we're going to call these lymphatic capillaries lacteals. And the third function of the lymphatic system is to provide immunity or to protect the body against infection. And it allows us to live in the real world along with other organisms. So when we talk about the immune system, many cells of the lymphatic system, they're going to provide defenses against disease and permanent immunity against future infections. So we'll learn that we have these memory cells. We get infected with a bacteria or a virus or something like that. And, we're going to fight this current infection. And, it might take us 10 days or 7 days or it'll take us a while to start feeling relief from that virus. and it is going to take our body a while to get adjusted to that new virus that's in our body, and we have to make those cells that are going to fight off that current infection. But what the body will do is make these things called memory cells that will protect us against future infections. So if we ever encounter that same virus or bacteria again, we already have these memory cells that will kind of pick up and say, hey, we've been exposed to this virus or bacteria before, and we don't have to relearn what this virus or bacteria is. We already have the defense mechanism to kind of kill it off. And so that's what the body will do, is instead of waiting that 10 days to feel better, we'll start feeling better in 3 days, 4 days, 2 days, 5 days type thing. And then we'll see that the aging of the lymphatic system begins before birth. So your lymphatic pathways. If we look at, this picture, and I'm advancing the slide a little bit, so you can see how the lymphatic system is kind of really intertwined with this capillary system or the cardiovascular system. And, this is the site where, we had that slide that we'll re-look at that has the, I'll just show it to you, that slide. That we've seen before. We will relook at this slide, but this is where we have that 20 liters and that 17 liters with an overall 3 liters. So we're we're looking at kind of like this space right here, and this capillary bed. We have the arterial end and the venual end. So we are going to kind of pick up this fluid, this interstitial fluid, and we're going to pick up the fluid from the interstitial space, and it's going to go in these lymphatic capillaries. And then once that fluid goes into the lymphatic capillaries, what we'll see is that they'll go into like these larger structures called lymphatic vessels, and then they're going to merge into these things called lymph nodes, which that's what you feel when you might have an infection, you might have swollen lymph nodes, and then they're going to go into these larger lymphatic vessels. Then we have these lymphatic trunks, lymphatic collecting ducts, and then eventually they're going to go into your subclavian veins that are in your thorax, and then eventually they're going to go back in the blood plasma and back into the heart and mix with your circulatory system. So this extra 3 liters of fluid on average is going to eventually go back to the circulatory system. So let's see, I actually should, so from the collecting ducts, we'll just go ahead and underline that. So you'll need to know that. So these lymphatic capillaries, so we're kind of talking again, these things right here, the lymphatic capillaries. The lymphatic capillaries, they're closed-ended tubes. You can see that there's, they're very closed. They don't have spaces. And we'll look at this more on a microscopic level later on. They parallel blood capillaries, which we see that, we see that they are very intertwined with the cardiovascular system. They have very thin walls. Remember, in order to do like gas exchange or fluid exchange, we like to have that simple squamous epithelium so that things only have to cross one layer, that simple layer, one layer, in order to go from one side to the other. And we call that a simple squamous epithelium, or we call it endothelium. So this tissue fluid, or the interstitial fluid, or the extracellular fluid, so this interstitial fluid is going to enter the lymphatic capillary. So the fluid is going to go from, on the arterial end, we're going to push out fluid from the arteries, and then that fluid is going to get picked up by the lymphatic capillaries. So the interstitial fluid enters the lymphatic capillaries. And once that interstitial fluid enters the capillaries, those lymphatic capillaries, we now call it lymph. We don't call it interstitial fluid anymore. We call it lymph. And then we also see that the lymphatic capillaries drain your cerebral spinal fluid. And then we also know that it transports the lipid-soluble substances to the bloodstream via the liver once the liver packages those. And we know that the lymphatic capillaries merge into lymphatic vessels, which we saw up here. All right, so here we can see that we're going to rehash this a little bit. The arteriole and the venules divide into smaller branches called these blood capillaries that form this complex loop of network in the tissue cells, and then these lymphatic vessels divide into smaller branches called lymphatic capillaries that kind of weave between the blood capillary and draws that interstitial fluid into it. And we'll talk about how that happens later on. And remember that these lymphatic vessels or these lymphatic capillaries are closed-ended, narrow tubules. These lymphatic capillaries are very thin-walled, and lymph is going to flow from the lymphatic capillaries, and eventually are going to go back into the circulatory system. All right, so here we can see a microscopic view of a lymphatic vessel. Like I said earlier, that the walls are going to be very similar to veins, but they're going to be a little bit thinner. And you can see like a little valve here. And that's just going to prevent backflow of the lymph to prevent backflow from going backwards. So your lymphatic vessels are going to have 3 layers. We have an inner layer, a middle layer, and an outer layer. Your inner layer is going to be made up of an endothelial lining, whereas your middle layer has a bunch of smooth muscle and elastic fibers. Your outer layer is just connective tissue. And we have the semilunar valves that are going to be similar to like your aortic valve or your pulmonic valve. And these larger vessels are going to lead to your lymph nodes and then to your lymphatic trunks. So remember how we have like skeletal muscle contraction that allows, that helps that venous blood flow. You contract your skeletal muscles, and the venous blood flow kind of helps. Skeletal muscle contraction helps venous blood flow go back to the heart. Well, the same system or the same concept applies for lymph as well. Skeletal muscle contraction also allows that lymph to get back to those subclavian veins to get returned to the heart. So when we get to the lymphatic trunks, so remember that we have lymphatic vessels, lymph nodes. Now we're talking about the lymphatic trunks. So when we get to the lymphatic trunks, that's where the lymphatic trunks drain from the lymphatic vessels, and they're going to be named for the regions that they serve. So you can see the lumbar, intestinal, intercostal, bronchial, blah, blah, blah. So the lumbar trunk is going to drain from like the lower limbs, lower abdominal wall, pelvic organs. The intestinal trunk is going to drain the abdominal viscera. The thoracic duct drains lymph. We'll save that one for a second. All right, so once we have those trunks are going to drain into these things called lymphatic collecting ducts. And we have 2 collecting ducts. We have a thoracic duct and a right lymphatic duct. So your thoracic duct is going to be the longer and wider of the 2 ducts. It's going to drain the majority of the body. It's going to begin at this thing called the cisterna chili or the cisternic hyli. People pronounce it different things, and it's going to empty into your left subclavian vein. It's just in a large sac at the end of the thoracic duct. So you can kind of see it right here, right here. That's your cisternic hyli. Or chili, I've heard it pronounced different ways. But then your right lymphatic duct, it's going to be smaller than your thoracic duct. It's in your left thorax, and it's going to drain the right upper portion of your body. So your thoracic duct is going to drain lymph from the intestinal, lumbar, and intercostal trunks, as well as from the left subclavian, left jugular, and left bronchomediastinal trunks. And your right lymphatic duct will drain the right thorax, your upper limb, head, and neck. Okay. All right, so back to this picture. So we're just going to do a little review of this picture. So remember on the arterial end, so arterial end, E-R-I-O-L-E. And then over here we have the venule end. And remember that in here we have a bunch of different proteins that are going to be in our blood. And remember, proteins, large proteins, do not get filtered out of the blood. They don't get pushed out of the blood. And if you also remember, protein pressure is called osmotic pressure. It's called colloid pressure. It's called colloid osmotic pressure. We can also call it protein colloid osmotic pressure. Everything has 47 different names. So we call it, basically, if you hear osmotic pressure, think of protein pressure. Always doesn't mean always, and never doesn't mean never, but for simplicity's sake, when we say osmotic pressure, think of like pressure coming from proteins. Now, I guess I should say pressure coming from large proteins that aren't filtered out. Now, if we have really tiny proteins, if we have those really, really, really tiny proteins, those really tiny proteins could be kind of thrown out along with that along with that fluid that gets pulled out. So on the arterial end, we have fluid that's in our plasma, and so we have and all of this is extracellular fluid that's in our that's in our blood. And then we also have fluid here inside of our cells, and we have fluid that's kind of out here in this space. So when we have water-based fluid, we call that hydrostatic pressure. So we have water fluid called hydrostatic pressure, and we have protein pressure. So those are the two pressures that we're going to be dealing with. And so this water pressure or hydrostatic pressure is going to be what pulls water out of the arterial end. And so if we look at this hydrostatic pressure, the pressure out here is going to be greater than the pressure on the inside. So we're going to be pulling, the hydrostatic pressure is going to be pulling water out. And on the arterial end, we say that is going to be about 20 liters per day. Now, because we've pulled all of this water out, once we get to the venule side, we don't have as much water. So now that hydrostatic pressure is not as great on the venule side. And you can see that the numbers here have really decreased from 35 to 16. And because large proteins don't get filtered out, our protein pressure is going to remain the same. And then if we kind of subtract the numbers, we get 35 minus 24 is 11. 24 minus 16 is 8. But you kind of have to look at which number is larger. And on the left side, since our 35 is greater than 24, our hydrostatic pressure is greater than our protein pressure. That's why we're going to push water out. So we're not, I guess I shouldn't say push water out. What I should say is that because of this, the pressure is greater on the. I guess I should say that we are drawing water out of the arterial end as opposed to pushing water out of the arterial end. So I should say we are pulling water out of the arterial end. And on the venual end, because the protein pressure is greater than the hydrostatic pressure, we are pulling water back in. So we are going to pull water back in from the interstitial space back into the vessel, into the venual end, to kind of help counteract or counterbalance that protein pressure. And we'll see that on average it's about 17 liters per day. So 20 liters minus 17 liters is going to be about 3 liters. So that 3 liters is what's going to end up going, hopefully, into that lymphatic system. So the driving force for lymph formation is going to be the hydrostatic pressure that is in the tissue fluid. All of this pressure here, this hydrostatic pressure that's in this tissue fluid, is going to be the driving force for the for lymph formation, or I guess we could say for interstitial fluid formation, or tissue fluid formation, which eventually will become lymph once it enters the lymphatic capillary. So, all right. So tissue fluid and lymph. So lymph is going to depend on tissue fluid formation. That's kind of what we just went over. So hydrostatic pressure, that water pressure, is going to force fluid out of the blood plasma because that 35 is greater than 24. Our water pressure, our hydrostatic pressure, is greater than our protein pressure. So here we have that plasma colloid osmotic pressure. So our protein pressure is going to be greater on the venual end. Our hydrostatic pressure is greater on the arterial end. So we can say that we are going to filter 20 liters of water out from the plasma, and then we can reabsorb 17 liters on the venual end, and then we're going to be left with 3 liters of tissue fluid. So really, what is tissue fluid? So tissue fluid is going to have the same composition as blood plasma, with the exception of large proteins, because remember, large proteins do not get filtered out of the blood. Large proteins contribute to that plasma colloid osmotic pressure, so they're not going to get filtered out. Small proteins can get filtered out, but not large proteins. So those large proteins are going to remain in the blood. So tissue fluid is basically going to be blood plasma without your large plasma proteins. Now here you can see that, tissue fluid can contain small plasma proteins like albumin. So in my drawing, I have these really, really, really tiny proteins here. So these really tiny proteins can escape through the holes they can follow through hydrostatic pressure, but they have to be really, really tiny in order to do so. And white blood cells can be in your lymph, but red blood cells do not leak out. So your red blood cells are going to stay in your plasma, whereas out here you can find some white blood cells. And it's not necessarily that white blood cells leak out, we just have white blood cells kind of free floating out here. And then we also know that, that tissue fluid or what leaks out is going to contain water because that's our hydrostatic pressure. We're pulling water out. And then, because of diffusion concentration gradients, we might have nutrients and gases and hormones that are either going to, one, diffuse out because of concentration gradients, or are already going to be free floating in this hydrostatic space or in this interstitial space. All right, so now we know that we have an increased hydrostatic pressure on the arterial end, and we know that we have an increased plasma colloid osmotic pressure on the venual end. And we know that because of those two pressure differences, we know that the hydrostatic pressure outweighs on the arterial end is going to outweigh the colloid osmotic pressure on the venual end, which is why we get that net of 3 liters per day. So really, we could say that tissue fluid is going to be extracellular fluid, and we also call that interstitial fluid. And remember that once that interstitial fluid enters the capillaries, or that lymphatic capillary, once that fluid enters that lymphatic capillary, it's going to be called lymph. If the lymphatic system did not pick up that interstitial fluid, we could lead to excess tissue fluid or excess extracellular fluid or excess interstitial fluid we call that edema. And we see that, here's an example here, we see that, with breast cancer, if we remove the breast and maybe the cancer has spread to your axillary lymph nodes, and you remove those lymph nodes as well, you're going to remove portions of that lymphatic system that can't pick up that interstitial fluid. You're going to see edema around that area, and you might have to wear a compression sleeve or a wrap to help control that edema. But your arm, your patient's arm, could be edematous. Edematous is the adjective or the adverb form of edema. So you could see edema of the arm, or the patient's arm could be edematous because we have removed the lymphatic system or the axillary lymph nodes in that area. All right, so lymph flow. So again, we're still talking about pressure changes when we are looking at the interstitial fluid or the interstitial space versus the lymphatic capillary. Lymph is going to go from the interstitial space to the lymphatic capillary because of hydrostatic pressure. So the lymph inside the lymphatic vessels have low hydrostatic pressure, just like the venous blood does. And how does that lymphatic flow happen? We talked about muscular contraction. Remember, contraction of skeletal muscle compresses the venous system and helps move the blood, but it also does that with the lymph as well. When we take deep breaths and we breathe out, those breathing out, that breathing out mechanism is going to create that low pressure in the thorax or your thoracic cavity, is going to help move the lymph. The high pressure in the abdomen for inspiration is going to help send the lymph from the abdomen to the thorax. And then we also have, your smooth muscle that is in the lymphatic vessels. They can contract to help aid in the flow of lymph. And then we already said that valves are in the lymphatic system to prevent backflow. So now we have valves in your heart, valves are in your veins, and valves are in the lymphatic system. And we'll see that lymphatic flow is going to be highest during physical exercise, which makes sense because when we do strenuous exercises, chances are we are moving our skeletal muscles a lot. All right, so lymph function. So what do the lymphatic capillaries do? We mentioned earlier that it absorbs those dietary fats in the small intestine or in the digestive system, specifically the small intestine. And we say chylomicrons, and we're going to deliver them eventually to the bloodstream. So we transport the lipids to the liver, and I said those are called lacteals. So these chylomicron things, they can't diffuse into the capillaries from the small intestine, and this is where the lymphatic system comes into play. The lacteals are going to pick up these chylomicrons because those fats or these chylomicrons are lipid soluble. Remember, fats and water, they don't mix. And then also these small proteins or hormones can be filtered by blood capillaries and go to the bloodstream, but also transport foreign particles to the lymph nodes. That's another function of lymphatic capillaries. Remember that we deliver that excess fluid, that 3 liters, back to the bloodstream via the subclavian veins. Delivery of foreign particles to lymph nodes, I kind of already said that. So we'll just get rid of that. So that's going to help protect the body against infection. And then we know we have these, we haven't really talked about these flap-like valves that are between the cells. We'll see a picture of these in the next slide, but this is how lymph gets in, or the interstitial fluid gets into the lymphatic capillaries. These valves are going to be pushed inward when the hydrostatic pressure is greater on the outside of the lymphatic capillary, but closed when the hydrostatic pressure is greater on the inside of the capillary. All right, so what does that mean? So we'll look at this slide. So here we have the end of the lymphatic capillary, and you can see we have these little, this all blue space is just a bunch of connective tissue, or basically your interstitial space. This is all your interstitial space, and you can see your simple squamous epithelium here. Remember, that's your one layer, and you can kind of see that these cells are overlapping a little bit. And that they overlap, and they almost form these valve-like things. And when the hydrostatic pressure is greater on the outside versus the inside, that water pressure is going to push the interstitial fluid into the lymphatic capillary. It's going to open up little valve things, or these joining cells, and allow fluid to get pushed into the lymphatic capillaries. And then when the hydrostatic pressure is greater on the inside of the lymphatic capillaries, that pressure is going to push these kind of cells kind of back together to create that little valve being closed, so that water or that lymph cannot go back out into the interstitial space. All right, so this tissue fluid or this interstitial fluid just goes into the lymphatic capillaries between these epithelial cells because of increased hydrostatic pressure on the outside or in the interstitial space. Remember that I said earlier that hydrostatic pressure is the driving force of lymph formation. So again, we're still talking about hydrostatic pressure. All right, so lymphatic tissue, lymphatic organs. So lymphatic tissue has, different cell types. We have lymphocytes or lymphatic cells. We have these things called macrophages. Macrophages, think of like phagocytes. P-H-A-G means to eat. Macro is large. So here we would say large eaters. And lymphocytes and macrophages are responsible for immunity. They're going to be the ones out there that are going to gobble up a lot of your cellular debris, a lot of your viruses, bacteria that have been killed off. We also have MALT. It's called mucosa-associated lymphoid tissue, or MALT. And we're going to see these in your digestive tract, your respiratory, urinary, reproductive tract. Basically, it's unencapsulated lymphatic tissue that's going to help us help protect us against pathogens. And remember, a pathogen is any disease-causing agent. So think of like your tonsils, your appendix. It's just a cluster of lymph tissue. Basically, these are clusters of lymphatic nodules. We also have these things called Peyer's patches in your lymphatic nodules or your lymph nodes. You'll also see these in your ileum, which is going to a part of your small intestine. They're going to contain immune cells. We talked about macrophages. We'll talk about dendritic cells, T cells, B cells. And these Peyer's patches, these cells, they're going to respond to infection. We'll talk about the antigen antibody reaction, and when we talk about the immune system and how cellular immunity versus humoral immunity or cell-mediated immunity. I can't talk. Cellular mediated immunity. Cell-mediated immunity. There we go. Or humoral immunity. All right, so lymphatic organs, we have the lymphatic tissue, lymph nodes, thymus, and spleen are going to be the big ones that we talk about. All right, so lymph nodes. They're usually like bean-shaped, less than 2.5 centimeters long. They're tiny. They're about that big. And they're going to be located along lymphatic vessels. And they're like filters. They're going to be small filters, and they're going to filter out those pathogens from the lymph itself. You're going to see a lot of your B cells and T cells. They're going to live in the lymphatic tissues until they're in the lymph nodes. Your B cells and T cells are going to live in your lymph nodes until they are going to be called to action And so your lymph nodes, they contain these lymphocytes. Lymphocytes, remember, are it's a type of white blood cell. So those are going to be your B cells and your T cells to attack viruses, parasites, bacteria. Your lymph nodes have these macrophages, remember, phagocytes that engulf and destroy any type of foreign substances, cellular debris, or damaged cells. We're going to find that lymph nodes are found in groups or chains along paths of larger lymphatic vessels throughout the body. We'll see that in a couple of slides. And then we'll see that lymph nodes lie within those regions or groups of chains, and I guess I should end in that one. But lymph nodes are not found in the central nervous system. So here we can just see a portion or just like a lymph node. So here, I guess I'll look at, here you can just see the lymph node with your lymphatic vessels. So here you can see like the outer layer, this, we call that a capsule. And then you can see these things called germinal centers. The germinal centers is where B cells or B lymphocytes are going to proliferate. And then we have this inner portion called the medulla. That's where your T cells are going to be. And your macrophages. So all up in here, those are going to be your T cells and macrophages. That's where these are your germinal cells. Those are going to be like where your B cells proliferate. Another thing that I'll mention, these are afferent lymphatic vessels. Remember, afferent, if you remember from the nervous system, afferent means to go into, efferent means to go out of. So we have like an afferent arteriole and an efferent arteriole. When we talk about the kidney, we'll talk about the difference between those two, but an afferent arteriole would be something that goes into, an efferent arteriole would be something that goes out of. Like I said, we'll talk about that when we talk about the glomerulus and the kidney. But we also have this thing called a hilum, and that's going to be like right here. A hilum is basically an area where blood vessels or nerves or something like that go into, in this case, a lymph node. We have a hilum for like the lungs. We have a hilum for the kidney, and basically that's where the nerves and blood vessels go into the lung or the kidney or the lymph node. And we can see that lymph flow is one direction. It goes in the afferent side. Remember, afferent goes into, and it goes out the efferent side. Remember, efferent means to go out of. I already mentioned the hilum. So the hilum is just the region of the lymph node through which blood vessels and nerves pass. Let's see, that's about it for this slide. All right, so here you can see different regions of lymph nodes or different lymph nodes in your body. Those are the little bulges that you see like up here in the cervical or the axillary region, or your thoracic lymph nodes, your inguinal lymph nodes, your pelvic lymph nodes. So your cervical lymph nodes are going to drain, are basically they're in the neck, and they're going to drain lymph from the scalp and the face. I guess those are your cervical. Your thoracic lymph nodes are in the thoracic cavity. Your axillary lymph nodes are going to be in your armpit. We have these things called supratrochlear lymph nodes. They're in the medial side of the elbow. The medial side of the elbow. Remember anatomical position. Abdominal lymph nodes are in the abdominal cavity. Pelvic lymph nodes are going to be in your pelvic cavity. Inguinal lymph nodes are going to be in your inguinal region. And your inguinal lymph nodes, so basically your lymph nodes are going to uh, cover the area that's kind of closest to it. So, if you have a cut or something in your big toe, or let's say you have a bacterial infection in your knee, the closest pathway to the lymph node would be, or the closest lymph node would be your inguinal region. So if you have a bacterial infection in your knee, your inguinal lymph nodes would swell or enlarge because of that bacterial lymph nodes. It's not like if you have a bacterial infection in your knee, your cervical lymph nodes would be affected. Your inguinal lymph nodes would be affected. All right, so functions of lymph nodes. We have two primary functions of lymph nodes. I'm going to filter out harmful particles or potentially harmful particles from the lymph, and we also provide they also provide immune surveillance. They're going to monitor the body fluids. Remember macrophages and lymphocytes. And remember, anytime that I say lymphocytes, you should be thinking of B cells and T cells. And remember, I haven't said, we haven't talked necessarily about memory B cells and memory T cells. But I did mention them, way up front about, repeated exposure to the same virus or bacteria. So kind of put that in the back of your brain that your B cells and T cells are going to be kind of living in your lymph nodes. And remember your germinal cells and the medulla of your lymph nodes that we talked about up here. All right. So, lymph nodes, along with the red bone marrow, lymph nodes are centers for lymphocyte production. So your lymph nodes also produce lymphocytes. And these lymphocytes are going to, remember lymphocytes, B cells and T cells are going to attack those pathogens in the lymph nodes. We have those macrophages that are going to act as phagocytes that engulf, destroy foreign substances, cellular debris, damaged cells, stuff like that. Okay, so lymphedema. We briefly discussed lymphedema, or we've discussed edema as it pertains, that extra 3 liters of fluid that doesn't get into the lymphatic system. But we have this thing called lymphedema, and it's just basically a fluid accumulation because of a disruption of lymph flow, and it's usually because the lymph nodes have been surgically removed. So that example that we talked about several slides ago here, because of that breast cancer, would be lymphedema. All right, so how do we treat this lymphedema? Regular exercise. I talked about wearing that compression sleeve, raising your arm above the level of the heart. Gravity will actually help pull that fluid back down to your heart. We also have these things called compression pumps that will help drain the excess fluid. All right, your thymus. Your thymus is actually where am I? It's a soft gland. It has 2 lobes to it. It's going to lie in your mediastinum, more like at the upper portion between your lungs, kind of like right here in the upper part of your chest. It has different lobules, and those lobules contain these things called lymphocytes. And if you remember from that, the pathway of stem cells that we saw in a previous chapter, they're going to come from these things called progenitor cells that are in the red bone marrow. And most of your thymus cells or your these thymus cells are called thymocytes, and most of your thymocytes are going to be inactive. Now, so your thymocytes are going to be inactive. However, we do know that your thymus does produce these things called thymosins, and that's going to be those hormones that are going to be produced by epithelial cells in the thymus. So how do the thymocytes get activated if they are typically inactive? So your thymosins are going to stimulate the maturation of thymocytes into your T cells or your T lymphocytes. And at that point, they're going to leave the thymus to provide immunity. And the interesting thing about your thymus is it's large in infancy and early childhood, shrinks at puberty, and it's small in an adult. And once you are considered elderly, a lot of your thymus is actually replaced by adipose tissue or other connective tissue. There's a condition out there called DiGeorge syndrome. It's D-I-George syndrome. It's a chromosomal disorder that can result in an underdeveloped thymus. So if you have an underdeveloped thymus, chances are you're going to have an underdeveloped immune system. And if you have an underdeveloped immune system, you're going to have a tendency for frequent infections. All right, so here you can see the thymus. Here is where your thymus is. And you can see that a thymus in fetus is relatively large compared to a thymus in the adult. All right, so your spleen. All right, so your spleen is actually an elongated, oblong-shaped organ with has a splenic artery and a splenic vein that is divided into smaller branches, and it penetrates from the interior surface. The spleen has this capsule made of dense and fibrous covering that surrounds the spleen. The fibrous connective tissue extends from the capsule to the interior of the organ. We have these splenic sinusoids or capsules that are very thin vascular channels, and they're surrounded by a framework of reticular fibers. And the spleen also has this red pulp and white pulp, and the red pulp consists of a network of vascular channels called splenic sinusoids, and the red pulp extends throughout the spleen and surrounds the white pulp. The white pulp contains clusters of lymphatic tissue known as lymphocytes. The venous sinus is the large and first vessel in the spleen and makes up most of the entire red pulp. Let's see. Spleen is also your largest organ. It lies in the left upper part of the abdominal cavity. It's divided into lobules. Contains a lot of venous sinuses, said before, highly permeable, has a lot of holes in it that the blood can kind of get into and kind of get filtered out. I mentioned the red pulp and the white pulp. The white pulp has a lot of lymphocytes. Lymphocytes are, white blood cells. Remember B cells, T cells. Red pulp, as the name suggests, it's red, lots of red blood cells. But it also does contain lymphocytes, B cells, T cells, and macrophages. Remember, macrophages are phagocytic. Your spleen is, remember I said highly vascular, contains a lot of blood and does a lot of filtering out of stuff. So it filters out blood just like the lymph nodes are going to filter out the lymph. And your spleen, when your red blood cells, remember your red blood cells don't go through mitosis because they don't have a nucleus. So when your red blood cells are ready to die, they are going to die and they're going to come to the spleen to get broken down. And here, the macrophages are going to destroy, engulf and destroy foreign particles, as we mentioned before. We know that lymphocytes help provide protection. And then, if you've ever heard of Epstein-Barr virus, it's responsible for mononucleosis. I'm sure you've heard of that one. This virus actually attacks B lymphocytes and uses the B lymphocytes to replicate itself. When the immune system manages to halt the Epstein-Barr virus production, the virus still remains dormant in these B cells for the rest of the life of the organism that it's staying in. So if you contract mononucleosis, this virus is going to remain dormant in your B cells for the rest of your life. And remember that B cells are also going to be found in the spleen, just like they are going to be found in the germinal cells of the lymph nodes. All right, so here's just a summary slide of the different organs of the lymphatic system. We have our lymph nodes, our thymus, and our spleen. Okay, so lymphatic system defends the body against pathogens. And remember, pathogen is just any disease-causing agent. It could be a bacteria, a virus, it could be a protozoa, it could be a fungus, it could be a parasite, anything that causes a disease in the body. So viruses usually have a simpler structure than living cells. They are not organisms by themselves. Viruses have DNA or RNA in their makeup, and they also have this thing called a protein coat. Think of like herpes virus or COVID virus, HIV, human immunodeficiency virus. What makes viruses interesting is viruses cannot replicate by themselves. Typically pathogens can replicate by themselves, but viruses cannot. Viruses have to use another source. So they're gonna, viruses actually go into our cells and use our DNA and our RNA to replicate themselves. Bacteria can be single-celled organisms, and they can cause infections such as like staphylococcus or streptococcus. Like if you have strep throat, that would be a strep-causing organism. All right, so immunity. Remember, immunity is just how our body prevents either pathogen entry or how we destroy pathogens that enter our body. We have two different ways our body fights protection, or our body provides protection. We have these innate defenses and we have these adaptive defenses. Innate defenses are more general. They're going to work more immediate. They're non-specific. They're kind of like, you see a snake in your house and you blow up your house because you don't like snakes. That would be an innate defense. Whereas an adaptive defense would be you see a snake in your house, you figure out what kind of snake it is, and you take specific measures to get rid of that specific snake. That would be an adaptive defense. So an innate defense would be something that is going to take care of something very general, something very broad, whereas an adaptive defense would be something very, very specific. So general defenses, inflammation, phagocytosis, a fever, a cough, enzyme and tears, mucus. We'll talk about these, but these innate defenses are going to help us provide or protect us against a lot of different types of pathogens, different proteins or viruses or bacteria, fungi, parasites. We don't necessarily know the exact type of virus that we're talking about, or the exact type of virus that, or the exact type of bacteria, or the exact type of fungus or protein or parasite. We just know that something foreign has entered our body or trying to get into our body, and we're going to protect against it. So if you think of like your stomach acid doesn't care what type of protein it is. Your stomach acid is going to degrade the protein. Now, for adaptive defenses, that's where we're going to figure out what specific type of virus or body it is, and we're going to use our B cells and our T cells to counteract that specific type of virus, and we're going to produce those memory cells to provide future immunity. So let's say you get a splinter. Your body's first reaction is going to be to activate those innate defenses. You're going to, you get a splinter in your hand, you're going to get inflamed. You're going to have the phagocytosis, you're going to get pus. Your body's going to trap that bacteria. The mucus is going to form, and phagocytes are going to come in and kind of eat away at that cellular debris and get rid of, you're going to pull the splinter and your phagocytes are going to come in and eat up that gunk, and hopefully you're done. All right. So with innate, the body doesn't know what type of bacteria. It just says, we're not going to let it in. The body doesn't know. All right, so, we have this thing called species resistance, where certain species can get a disease and other species cannot. So humans can get measles, other animals cannot. Certain animals can get calcivirus, certain animals cannot. Canine distemper, that's in dogs. People cannot get canine distemper. So certain species are going to lack the receptors for that disease, or we might be at a different temperature than another animal, or we might have a different internal chemical environment that prevents that virus or bacteria from being able to grow inside of our body versus another animal's body. So that's species resistance. Mechanical barriers. We consider mechanical barriers the first line of defense for the innate defenses. So whenever we have any type of bacteria or virus, the mechanical barriers are the first line of defense. They are going to prevent the entrance of pathogen into our internal body. So think of like your hair. Your hair forms a barrier. Your skin is forming a barrier to prevent pathogens from entering our body. Your mucus, you breathe in dust, you breathe in bacteria. Your mucus, your mucosal linings are going to prevent that bacteria from getting inside. You breathe it in, and hopefully you cough it out or sneeze it out, or that mucus is going to bring it back up to your nose, and you blow your nose and you blow out that bacteria. Your tears. Your tears have enzymes in it that are going to kind of get rid of that bacteria. Saliva, we have, enzymes in our saliva. So some of the examples here, as the epidermis, we know we have the 5 layers of the skin, and we brush off that stratum corneum layer, and it's going to remove some of that superficial bacteria. You wash your skin, you wash your hands after you go to the bathroom, hopefully. You're washing off that bacteria. You go touch a patient that's infected with something, you wash your hands to prevent transmission of bacteria from one patient to the other. We have ciliated epithelium. Remember that cilia is going to trap that respiratory gunk that you just breathed in, and hopefully you can exhale it or sneeze it out or cough it out or blow away. Your hair is going to trap different pathogens, and then you wash your hair. Tears, saliva, urine is going to help wash away any microorganisms. We have one thing that can, an example of bypassing this mechanical barrier is this thing called H. pylori or Heliobacter pylori. That's going to be kind of like an ulcer. That bacteria is going to get into your stomach, and it's going to burrow into the mucosal lining of the stomach. It's going to degrade it, and it's going to form a hole in your stomach, causing an ulcer. And then you're going to have to be on some, probably some antibiotics and maybe some type of gastric acid blocker, a proton pump inhibitor or something like that to prevent some gastric secretions to help heal that gastric lining. That would be an example. This H. pylori getting into your system would be an example of overcoming a barrier or overcoming that mechanical barrier. So remember, this mechanical barrier is your first line of defense. Now, the rest of what we're talking about, or what we're going to talk about for the innate defenses, are your second lines of defenses. So inflammation would be a second line of defense. Inflammation produces redness, swelling, heat, and pain at that local level. It's very localized. You get a splinter in your hand, your foot doesn't swell up, your foot doesn't get painful. So that redness is due to vasodilation or increased blood flow to that area. And that swelling is going to result from that vasodilation or that increased capillary permeability. And we're going to see that there's increased fluid in that space or in that tissue space. We have heat because of blood coming from deeper areas of that body. And remember, blood blood transports heat. We break down ATP and we create energy in the form of heat. So now that's why we have heat or localized heat. And then we have pain due to stimulation of pain receptors or nociceptors. And then with inflammation, we block off that infection site, so hopefully that infection doesn't spread to other areas of the body. We're going to see white blood cells that come in and destroy those pathogens by phagocytosis. We see that pus forms, and pus contains a lot of fibrinogen and other clotting factors that can form that fibrin network. We talked a little bit about that when we talked about the clotting cascade. And then we're going to have replace those dead cells. All right, so here is a summary slide of what happens during inflammation. Your blood vessels dilate, capillaries are more permeable, white blood cells come in, tissue fluid has clotting factors, fibroblasts, phagocytosis, and new cell division occurs. And you can see the results of all the actions that have been taken. All right, so another second line of defense is chemical barriers. So these are just chemical barriers that will help protect us against pathogens. So if we have, pepsin and hydrochloric acid in our gastric juice, are going to help destroy microorganisms. I mentioned tears before. Tears actually have an enzyme called lysozyme. That's going to help destroy microorganisms. Your salt, when you sweat, that salt is going to provide some protective mechanism. It's going to help destroy some microorganisms. We also have these chemicals called interferons. These are going to be produced by lymphocytes. Remember, lymphocytes is a type of white blood cell. So think of like your B cells and T cells. Also fibroblasts in response to viruses or tumor cells. So what interferons are, they're basically just proteins. Once the virus actually infects the animal cells, they are going to release these interferons, the cells are going to release the interferons that are going to inhibit viral replication. And they're also going to let the surrounding cells know that, hey, I'm infected with a virus. So, be on the lookout. So they're also going to protect nearby cells from being infected. And these interferons also stimulate phagocytosis. So they enhance the activity of other cells that can help resist infection and tumor growth. Another thing that for chemicals is defensins. And defensins are just peptides that are produced by neutrophils and other granulocytes. Remember, granulocytes, there's the white blood cells. There's granulocytes and agranulocytes. We'll see these in the intestinal epithelium, the urogenital tract, kidneys, and the skin. And basically, defensins are just going to make holes in bacterial cell walls and membranes, and they're just going to kind of cripple the microbes. So key takeaway, defensins are just going to kind of tear out the bacterial cell wall membranes. We also have collectins. They're just proteins, and they provide broad protection against some yeast and some viruses. What collectins do is they find the sugar molecules for all of these things, and they also look at structural patterns of these things to help identify the pathogen and make it easily phagocytized. And the last thing that we have is this thing called complement system. This complement system is basically a group of inactive proteins in the plasma and other body fluids, and they're going to respond to either an antibody-antigen reaction, which we'll talk about that with B cells, or the presence of foreign antigens. And once we have this binding, or once it's bound to the surface of that foreign cell, these inactive proteins are now going to be activated. It's going to initiate a bunch of cascade of reactions, which we're not going to cover in this class. But the result is that we're going to have inflammation and increased phagocytosis in the area, causing cells to lyse or to break up. So not only do we have enhanced phagocytosis, but we have the effectiveness of phagocytosis is going to be increased. So we're going to see increased phagocytosis, and we're going to see an increased effectiveness of the phagocytosis. And we'll talk a little bit about that when we talk about fever as well. So natural killer cells, it's very small population of lymphocytes. I told you to remember that, when I say lymphocytes, remember B cells and T cells, but lymphocytes are not only B cells and T cells. We also have these things called natural killer cells or NK cells that are going to provide adaptive defenses. And they're going to just defend against viruses, cancer cells. But they secrete these substances called perforins. We call them cytolytic substances that lyse the cell membrane or break up the cell membrane. So cytolytic, cyto means cell, or actually cyte means cell. And lytic, think of like lyse, L-Y-S-E, glycolysis or glycogenolysis. Lyse means to break up. And it's just going to help enhance that inflammatory process. All right, phagocytosis. We've kind of talked about phagocytosis several times now. Phagocytosis helps remove foreign particles from the lymph, um, and it helps in the tissues of the spleen, bone marrow, liver to remove particles from the blood. But the most active phagocytes or the most active phagocytic cells are neutrophils and monocytes. And if you remember, monocytes are the largest of the 5 white blood cell types. So monocytes will actually phagocytize larger molecules, whereas neutrophils digest smaller particles. So, all right, chemicals from the damaged tissue actually attract phagocytic cells. It's called chemotaxis. We'll talk about chemotaxis when we talk about specific, when we talk about the B cells and T cells and how they tag stuff and everything. But what happens with chemotaxis is that basically we tag these cells as injured cells, or that we say, hey, we have a foreign body, phagocytes, we need you to come and eat up these cells, and we're going to tag these cells. And then when we tag them, it's called opsonization. And when we do this opsonization process, these chemicals are going to get released, and that's called chemotaxis. And these chemicals are going to attract phagocytes to come kind of kill up these either injured cells or come kill off these, eat up these injured cells. So monocytes can also leave the bloodstream and become macrophages. So we have free cells, free floating in the blood, or they can become fixed. So when they leave the blood, so in the bloodstream, they are free cells wandering free in the bloodstream, but they can also leave the cell and go into tissues. At that point, they would be fixed cells. We have this reticuloendothelium system called a mononuclear phagocytic system that just consists of monocytes and macrophages in the body. All right, so we've seen this slide before. It's just a simple slide about phagocytosis. So I'm not going to review this slide in too much detail. We have the nucleus and the lysosome with the bacterium, and the bacterium kind of joins up with these receptors, and the cell engulfs the bacterium, and the lysosome will migrate up there, forming. So we have a phagosome, and once the lysosome hooks up with the bacterium inside, we have what's called a phagosome. And then down here we would have, the lysosome would dump its cellular contents in here, and we would those enzymes in the lysosome would kind of break up the cell membrane. We talked about the enzymes in the lysosome before. So, but what I wanted you to see is that all of these cells down here can act as phagocytes, macrophages, dendritic cells, monocytes, and neutrophils. All right. So also another innate defense. Remember, these are still our second line of defense. So a fever would be a second line of defense. You get a fever when you have a bacterial infection or a viral infection. So we see a fever when those lymphocytes are stimulated to proliferate. And proliferate just means to like replicate and begin to grow. So when we see the B cells and T stars starting to ramp up in our body, that's why we have the fever. And it's because of the substance called interleukin 1 or IL-1 that raises our thermoregulatory set point, which causes the fever. So our thermoregulatory set point goes from 98.6 up to, let's say, 101.5, and now we have 101.5 fever. So this interleukin 1, it's also called an endogenous pyrogen, or fire maker from within. Pyrogen means fire maker, and endogenous is within. And this elevated body temperature indirectly inhibits microbial growth. So what happens is basically like bacteria and fungi like to use iron for their replication process. And we know that iron is in heme. Remember hemoglobin, and that transports oxygen and stuff like that. So what happens is when we get a fever, our body naturally takes iron from our hemoglobin and puts it back into our liver and spleen. Because we don't have iron in our hemoglobin, our bacteria, the bacteria and fungi can't use that iron for normal replication process. So that's how that's what it means by indirectly inhibits microbial growth, because they don't have that iron for their normal replication process. And then several slides ago, I said that it increases phagocytic activity as well as increase the effectiveness of phagocytes. And I said we talked about that when we got to the fever slide. So this mildly elevated body temperature increases phagocytic activity as well as increasing the effectiveness of the phagocytes. So here is just a summary slide of what the different, remember, primary mechanical barriers are, and then all of these are considered secondary. Non-specific innate defenses. Remember, at this point, we don't care what type of virus or bacteria this is. We just know something foreign is in the body or trying to come in the body, and we're trying to do what we can to prevent it, either from coming into the body, or once it has come into the body, what can we do to stop it from spreading? Now we move on to our third line of defense. This is going to be a slower process. Our third line of defense is the body's going to have to know exactly what type of bacteria we're working with, exactly what type of virus it is, in order for it to counteract it. What virus are we dealing with? So it's our third line of defense. So first, what we have to decide is the body's going to look at that molecule or that potentially foreign substance and decide, is it part of the body, we say it's self, or is it non-self? Now, I'll back up a little bit. So how does the body distinguish molecules that are self from non-self? So remember that, some of this, the B cells and T cells, they're kind of formed, originally, stem cells and stuff like that are formed in the bone marrow. And, they have to be vetted before they can be released in the bloodstream. And we also have a whole bunch of molecules that are in the bone marrow as well, such as, insulin, hemoglobin, anything that our body's going to come in contact with are going to be in the bone marrow as well. So if a B cell or a T cell binds to that molecule in the bone marrow, that's going to be bad, because we don't want that binding, and the body knows that that should not happen. So the body's going to get rid of that B cell or that T cell. Now, and if that B cell or T cell doesn't bind to that good molecule, like the hemoglobin, the insulin, the glucose molecule, the thymosin, or that specific hormone, then that B cell or T cell is said to be vetted. And it can go to the bloodstream now and, go to the lymph node or whatever. And so now these B cells and T cells are recognized as self, or have the ability to recognize all of our self molecules. It knows that, our insulin that the pancreas produces is a self molecule. Our glucose molecule is a self molecule, or our hemoglobin is a self molecule. Our thyroid hormone is a self molecule. But a bacteria that is introduced into our body, that's a foreign substance. That is a non-self molecule. Now, if a B cell or a T cell that does happen to bind with a normal cell in the in the bone marrow and accidentally escapes, remember, if they bind in the bone marrow, that's bad. And if it happens to escape, that's how we get autoimmune diseases. And we'll talk a little bit about autoimmune diseases later on. So we have these B cells and T cells that recognize our normal self cells. And now we have something that is introduced into our body that the body recognizes as non-self. It's a foreign substance. Remember that we have these antigens and antibodies and stuff like that. So our cells have antigens that are on the cell. And these antigens are going to, these antigens are like receptors that are on the cell. And if there's like a linkage between the cell and that foreign cell, that's going to evoke an immune response, and that's going to result in the production of antibodies. And remember, antibodies are in the bloodstream. So antigens are on the cell itself, and the antibodies are in the bloodstream. Now we're going to start this whole immune system process, or the adaptive process, or the specific immunity response process. And that's going to be carried out by B cells and T cells and macrophages that can recognize specific antigens. So we have two different types of adaptive defenses. We have the humoral immune response, which is going to be more on the production of antibodies. So we have now an antigen-antibody reaction, and that's going to be more on the B cell side. And then we also have this thing called cellular immune response, which is going to be performed more by immune cells, and that's going to be more on the T cell side. And we'll talk about the T cell side first. I think that the B cell side, the humoral immune response, is easier to understand than the T cell side, but we'll talk about the T cell side first. All right, so here's just a summary slide of how pathogens enter in our body. Remember that our first line of defense is the mechanical barrier, and then our second line of defense are the chemical barriers and natural killers, inflammation, phagocytosis, fever, and then our third line of defense is cellular immune response, which is going to be more T cells, and our humoral immune response, which is going to be more B cells. And before we get into talking about the B cells and the T cells, we'll do a little bit of review on some antigens and talk a little bit more about antigen, antibody stuff. And some lymphocyte origin, stuff like that. All right, so remember antigens, those are the receptors on the surface of the lymphocytes. So on the surface of the B cells and the T cells. And they enable the cells to recognize substances or an object as non-self. So when we talked about blood typing, we had the blood type A and the anti-B antibodies. So they have the A antigen. If you have blood type A, you have the A antigen, but you have anti-B antibodies. So blood type A, you would have the A antigen, but you have anti-B antibodies floating around in your bloodstream. You have type B, so you have B antigens that are on the surface of the red blood cell, and then you have anti-A antibodies floating in your bloodstream. So remember, antigens are on the cell itself. And antigens can actually be, proteins, polysaccharides, glycoproteins, glycolipids. And if there is a reaction, they're going to trigger the production of antibodies. Now we have these things called haptens. It says here, haptens are small molecules that do not initiate an immune response by themselves, but when combined with a larger molecule in the body, they can evoke an immune response. So let's think of like poison ivy. So we have this thing called, it's spelled Urushiol, urushiol. It's a toxin that's found in the poison ivy, and when it's absorbed through the skin from the poison ivy plant, this urushiol undergoes oxidation in the skin cells to generate this hapten, which is going to react with the skin proteins to form this called hapten adduct, or this haptenic response. Now, typically your first exposure to poison ivy only causes sensitization, so you don't see any reaction, but your B cells are going to make antibodies to that hapten adduct. And so after a second exposure, those proliferated B cells can become activated, generating an immune reaction, producing, the typical blisters that you see of poison ivy exposure. Now, some haptens can produce or induce an autoimmune disease. So hydralazine, for those nursing students, that's actually a blood pressure medication. It lowers blood pressure, but it can occasionally produce a drug-induced like lupus type disease in certain individuals, lupus erythematosus. It can also this also appears to be the mechanism by which anesthetic. We used to use this anesthetic gas called halothane, which caused this life-threatening form of hepatitis. We used to call it halothane hepatitis. We don't use halothane anymore. You'll see it used in like third world countries, but halothane worked by this hapten as well. Penicillin could be a hapten. It could cause autoimmune hemolytic anemia, causing your red blood cells to lyse. All right. So here's this chart that I was talking about, earlier when I said, stem cells. We've seen this chart before. We talked about the granulocytes with neutrophils and the eosinophils, basophils. And previously we looked at, reticulocytes enter the circulatory system as erythrocytes. All right, so lymphocytes, they're produced throughout life. They start in fetal development. So the red bone marrow, remember that's where our red blood cells, white blood cells, platelets are produced. They start out as these things called lymphoblasts. So we're going to start kind of right here at lymphoblasts. And half of these cells are going to they're basically going to go to, they're either going to go to the thymus. If we go down this left pathway, they're going to go to the thymus to produce T cells or T lymphocytes, or they're going to stay in the bone marrow to become B cells. We go down this right pathway, and then eventually be released into the bloodstream. And remember that eventually T cells are going to be in the medulla of the lymph node, whereas the B cells are going to be in the germinal cells of the lymph node. So to remember where the lymphoblasts go, I remember as T cells are thymus. So here, thymus T cells. And I remember bone marrow, B cells, bloodstream. And then remember, oops, I didn't mean to draw that line, sorry. And then remember they both go to the lymph node where B cells, they're clustered in lymphatic nodules. B cells are going to be in the germinal cells. Actually, it's clustered within the lymphatic nodules. I'll just type it here, germinal cells. And then T cells are clustered, plus T cells are found in the medulla. All right. All right, so the two main types of lymphocytes, remember we have the natural killer cells, which are a type of lymphocyte. So we're not really talking about the lymphocytes, but or the NK cells. But so we're going to focus on the T cells and the B cells. So the T cells, remember those are from the thymus. Once we go from the lymphoblasts, we're going to go down to the thymus to T cells. And T cells are going to make up about 70 to 80% of your circulating lymphocytes. And you can see that they settle in your lymph nodes, thoracic duct, and then white pulp of your spleen. And remember, in your lymph nodes, your T cells are going to be residing in the medulla. Your B cells, remember your B cells come from the bone, differentiate in the bone marrow, get released in the bloodstream, and then they're going to be in the germinal cells of the lymph node, as well as the spleen, bone marrow, and intestinal lining. Here you can just see the steps of how we go from bone marrow to the bloodstream. And basically, this is just a recap of the previous slide. T cells are going to go to the thymus. B cells are going to get matured in the bone marrow and go that way. All right. Oh, and I didn't say, 20 to 30%. If this is 70 to 80%, this one's going to be 20 to 30%, the B cells. All right, just a summary slide of what we talked about previously. One thing I will point out for the primary function of T cells, for cellular immune response, which I know we haven't talked about yet, T cells are going to interact directly with the antigens or the antigen-bearing agents to destroy them. So T cells attach directly to cells displaying those foreign antigens, and they're going to destroy those foreign antigens. Oop. All right, so T cells and the cellular immune response. We also call it a mediated response. So these lymphocytes, before doing their thing, they're going to have to be activated. In order to be activated, they have to have this thing called an antigenic fragment is going to have to be attached to the antigen presenting cell. And we say, an antigen presenting cell, we call it an APC or an accessory cell. So these antigenic fragments, all they are just going to be processed fragments of the antigen. So it's kind of like, so far we're confusing. So it's right now, I'm very confused. So we have to activate these T cells. But in order to activate these T cells, we're going to have to somehow put these antigenic fragments on the antigen presenting cell. So what's going to happen is that these dendritic cells, remember dendritic cells are phagocytes, or one type of phagocyte. It's going to phagocytize that antigen via receptor-mediated endocytosis. If we go back to this slide, that's all we're doing here, is just doing phagocytosis. Receptor-mediated endocytosis means that we have a receptor here, and we are doing phagocytosis. We are phagocytizing this bacterium because we had that receptor that picked up that bacterium attached to that receptor. So that's receptor-mediated endocytosis. So these dendritic cells are going to phagocytize that antigen, that receptor by receptor-mediated endocytosis. They're going to process that antigen, and they're going to display that antigen on its cell surface. So let's say that that's going to be our dendritic cell. All we're going to do is just going to display that antigen right there on our cell surface. Now we call this cell an antigen presenting cell. We're going to find these in our epidermis, mucous membranes, and lymphatic organs. Now, or another example, we have a macrophage that's going to phagocytize that antigen, digest it. We have that phagolysosome that we saw. We're going to process that antigen. We're going to display those antigenic fragments on the cell membrane. So we're still going to follow, wherever I am. We're still going to follow this whole process. We're going to have this phagolysosome. Basically, what we're going to do is we're going to take a piece of this orange bacterium here. we're going to take a piece of that orange bacterium and we're going to put it on our cell wall. So we're going to have our cell, and our bacterium was orange. So now we're going to, let's say, and I guess I should make this one orange up here. There. So we've taken that bacterium and now we have it as orange. So we put it on there on our cell membrane. So now we have this antigen presenting cell. All right. So make sure. All right, so now these bacterial antigens are going to bind to these protein groups. We call these major histocompatibility complexes, or MHCs, that display these antigenic fragments on its own cell membranes, on the macrophageous cell membranes. We also call these MHCs, those complexes, HLAs, because they were first identified in white blood cells. We have two different types of MHCs. We have class 1 and class 2 MHCs. Class 1 are going to be on the surface of all body cells except for red blood cells, and class 2 MHCs are on the surface of antigen-presenting cells, thymus cells, and activated T cells. So these T cells are going to encounter these antigen-presenting cells that display these antigenic fragments bound to these MHC complexes. And now, once that T cell encounters the antigen presenting cell with that MHC protein, that T cell is now activated to protect the body against infection and provide cellular immune responses, or cell-mediated immunity. And T cells are going to secrete these things called cytokines that are going to enhance cellular responses to antigens. So if we look at, this picture, we still have the same picture, but then, we put that that piece of bacteria on our cell wall with our MHC complex. We've displayed the antigen, and then that T cell is going to come in and bind to that displayed antigen, that helper. And then we haven't talked about helper T cells, but we will. And that's going to proliferate once the cytotoxic T cell has bound to that displayed antigen with that MHC complex, or the major histocompatibility complex. The body is going to produce more T cells. And those T cells release those perforins. And then we'll talk also about memory T cells to live in the spleen and the lymph nodes. All right, so just to talk just briefly about these MHC proteins, remember that they act as the antigen presenting cells. We have the two different classes. Class 1, they're going to be found on all nucleated cells. So remember, red blood cells do not have a nucleus. Cytotoxic T cells can recognize and bind to class 1, forming that antigen complex. Class 2 MHCs. Class 2 MHCs, they're found on macrophages and B cells. And only these cell types can act as antigen presenter cells for the helper T cells. So once the helper T cells recognizes that foreign antigen, it's going to be activated. And then once activated, then we can do that cellular mediated immunity to secrete those cytokines, which is going to help or going to do that cellular response. Now, what are the types of cytokines? I just said cytokines. I mentioned up here, they secrete cytokines to enhance cellular response to antigens. And then I said, enhance cellular response to antigens. So what are cytokines? We have different types of cytokines. We have these things called colony stimulating factors that stimulate the bone marrow to produce lymphocytes. So remember, B cells are proliferated in the bone marrow. So colony stimulating factors cause B cells to grow and mature. They're also colony stimulating factors also activate macrophages. We also have interferons. Interferons are going to block viral replication, help macrophages to engulf those viruses. They're going to stimulate the B cells to produce antibodies. Interleukins, we talked about interleukins before with that IL-1, raising that thermoregulatory set point for fever. But they also control lymphocyte differentiation and proliferation. Interleukin 1 is going to activate T cells, whereas interleukin 2 causes T cells to proliferate. So interleukin 1 will increase your thermoregulatory set point and it activates your T cells. Interleukin 2 causes your T cells to proliferate or to replicate. Tissue necrosis factor is going to stop tumor growth. All right, so you're going to have to know, definitely know the types of cytokines. Okay, so we have different types of T cells. We have these things called helper T cells, and these helper T cells are activated when the antigen receptor binds to foreign antigens. So when we have that, when the antigen is going to bind to a foreign antigen. So when we have that, that bacterium binding to that antigen, we said that it secretes those cytokines. So secretes cytokines that enhances activity of other cells that helps the immune response. We talked about the cytokines here. And it also stimulates B cells. B cells are also called plasma cells to produce antibodies. Remember, we have that antigen-antibody complex or that antigen-antibody reaction, like agglutination would be one for red blood cells, like a transfusion reaction. And when we have a specific reaction, we have that it's going to be specific for that specific antigen. And then helper T cells also stimulate the activity of cytotoxic T cells. Cytotoxic T cells attack viruses that are inside your cells, and they also attack cancer cells. They are going to recognize the antigens on the surfaces near the MHC proteins. And they're also going to attach to cells that have those foreign antigens and destroy them. Remember, cytotoxic, hence the name cytotoxic. They release that perforin. Remember, perforin puts the holes in the cell membranes of the target cells. We also have memory cells that will help provide future immunity. And then we also have these things called regulatory T cells, which is going to suppress the immune response after we have defeated that pathogen. Because we don't want our immune system to keep working and working overtime after that pathogen has already left, because we don't want to develop an autoimmune disease. All right, so switching topics to the B cell and the humoral immune system. On the B cell side, this is usually when the virus or the bacteria has not entered the body yet. So B cells can be activated when that antigen and when that bacteria binds to that receptor. So if you have, type A blood and you're given type B blood, and those antibody antigen form that antigen reacts and we produce antibodies, that's what we're talking about with B cell. So we form that antigen antibody reaction. So those B cell receptors have been stimulated. Those B cells are going to require activation from those helper T cells. Remember, those helper T cells are going to secrete those cytokines. There we go, secrete cytokines. So we need those cytokines from those helper T cells. And those cytokines are going to help those B cells to proliferate. Remember those cytokines? I talked about those colony stimulating factors. Those colony stimulating factors are going to cause the B cells to grow and mature, and also to activate macrophages. So colony stimulating factors, releasing cytokines, and I'll put colony stimulating factors there. And the cytokines also attract those macrophages and leukocytes to the inflamed tissue. And the B cells are going to respond by basically enlarging the clone of identical cells. So what does that mean? We're going to go kind of down two pathways. Two pathways, which is going to kind of lead to four pathways, a total of four. So what we're going to do is we have this antigen antibody reaction. We're going to produce these memory B cells, which are going to provide future immunity. Those memory B cells are going to go to the lymph nodes, and they're going to reside in those germinal cells. But the other pathway is that those B cells are going to differentiate into plasma cells. We also call those effector cells. Remember, B cells are also called plasma cells, but now we're going to call them effector cells. And they're going to produce and secrete these large proteins, globular proteins called antibodies. Remember, those are in the bloodstream. We also can call them immunoglobulins. And these antibodies are going to be what is used to kill off those pathogens. And it says here that we can kill off about 200 per second per effector cell or per plasma cell. Now, how do we kill it off? Basically, those B cells are going to tag that pathogen as being foreign, saying, hey, we need you to come in and kill this cell. So when we tag it, we call that opsonization. So before it can be phagocytized, it has to be tagged. And we call that opsonization. And then the phagocytes can come in and kill this pathogen. Now, once we tag it, once we do this opsonization process, even though we haven't killed off that foreign pathogen yet, we've pretty much essentially rendered it useless. That tag, that opsonization process, it's rather large, and it's very hard for that pathogen to do its job because of that tag being so large. And now the pathogen can't really function properly. So essentially, it's that pathogen is now essentially useless because the tag that the B cells or the effector cells have put on the pathogen. And then now we've produced our antibodies in the blood, and it goes to the infection site. And so that's why we call it a humoral response, or an antibody mediated response. All right, so we have here we have our antigen antibody, our antigen antibody reflex or complex. And so we have the antigen reacting receptor hookup here, and then we have those cytokines from the T cell. And so basically what we're going to do is we're going to produce a bunch more of these B cells. And then we're going to form these memory B cells. And then we're also going to create a lot of these plasma or effector B cells. So these are going to provide immunity for now. And then these memory B cells are going to provide immunity later on. All right, so when we activate B cells, and this is just a picture form of what we've already talked about, we have the B cells, and then the B cells with the antigen receptor. And I think that, like I said, I think the B cells are easier to understand than the T cell side. So that's why I typically start with the B cell side. So the B cells combined with that antigen, we have that antigen receptor complex, and then we're going to proliferate and we're going to activate our B cells. But on the T cell side, remember, we have to engulf that virus, being with receptor mediated endocytosis. We form that lysosome, or that phagosome, and then we put that antigen, that displayed antigen, on the surface with the MHC protein. And then our cytotoxic T cells come in, and we have those helper T cells that are going to proliferate and release those cytokines. And then we have our interleukins. We create these cytotoxic T cells, and we're also going to produce memory cells for future and cytotoxic T cells as well. All right, and this is just a summary slide of kind of how we produce antibodies in B cells and T cells. This is just kind of a recap of what we've already talked about. I would spend some time looking at this slide, and I think it does a good job of synthesizing what we've talked about so far. All right, so antibodies themselves. So we talked about the antigens. Now we're going to talk about the antibodies. So we have these things called, these are basically globular proteins, gamma globulins or plasma proteins. We can also call them immunoglobulins. Basically, they are Y-shaped proteins of amino acids and side chains. You can see here that we have, kind of like basically from here, we have we call this inside the circle. Um, um, my mind's drawn a blank. Um, all right. Anyway, outside the circle, we have these things called light chains. So you can see here, this is a light chain, this is a light chain. And this is where our antigen is going to bind to the antibody. And we call this a variable region. That's what the word I'm thinking of, constant region. Okay, constant. So inside the circle, these are our constant regions. So this is going to be kind of pretty much constant for all of the antibodies out there. And then this area outside the circle, that's going to be kind of what makes the antibody unique for each different type of antibody. And you can see that we have, the heavy chains that are, more of the you can see the heavy chains versus the light chains. So we have two heavy chains and we have two light chains. Anyway, so the ends of the molecule, or the variable chains, the variable regions of the molecule, is what's going to bind to that antigen. And it's going to make it, that's going to, where we see that antigen binding site. We have 5 different types of immunoglobulins. I remember them so we have IgG, IgA, IgM, IgE, and IgD. I remember this, as GAMED, GAMED, as the order of smallest to largest. Just because, these are basically less than 1%. In reality, if you look at the percentages, a lot of textbooks will do GAMDE. So that even though these are less than 1%, this one is usually IgD is slightly more than IgE. So technically it should be GAMDE. But because this textbook does less than 1% of both, I just remember it as GAMED. All right, so with IgG, that's going to be your most abundant. You see 80% of your antibodies being IgG. That's going to be with, viruses and toxins and bacteria. These are going to be like your anti-RH antibodies. So, your anti-RH, that's your O positive, that's your blood type. So your anti-RH antibodies, that's your RH, your rhesus, that D antibody. IgA, it's found a lot in your exocrine gland secretions, so like your mother's milk, tears, your nasal fluid, gastric juice, intestinal juice, bile, urine. IgM, different foods, bacteria, also blood groups. IgE, exocrine gland secretions. You're going to see that with allergic responses. IgD is going to be found on different B cell surfaces. You're going to see that more in infants. Here's just a little summary slide. Here you can see that, they have IgD before IgE. So with antibody reactions, we have 3 different ways that antibodies can respond to antigens. The first way is basically a direct attack by antibodies. We'll see this with agglutination. And that's when we have the clumping of antigens. And that's going to make it easier for phagocytosis to happen. You have just a bunch of cells that are clumped together. Phagocytes can come in and just kind of eat at that clump. We also have precipitation. That's when antibodies bind to the antigen, forming this insoluble complex that can be easily phagocytized. If you took chemistry lab and you have stuff precipitate out, that's what we're talking about here. Neutralization is basically we're neutralizing a virus or a bacteria. We're going to basically cover up the harmful portions of antigens, essentially making them useless or harmless. The second way we can do it is activation of that complement system. It's more effective than the direct attack. We're going to see IgM and IgG antibodies. We'll see this with agglutination as well. Opsonization, that's when we tag that virus so that phagocytes can figure out which cell needs to be eaten up. And that's when opsonization is basically tagging that protein or tagging that virus or bacteria, where we'll tag it by coating that antigen with a bunch of protein, so it can be phagocytized easily. Chemotaxis, that's how macrophages and neutrophils are going to know what cells. They're almost like pheromones. They're chemicals that are released by the cells to say, hey, I need to be eaten up. So macrophages and neutrophils can figure out what to eat. Lysis, rupture of cell membranes of pathogens. Neutralization again, changing the structure of viruses as opposed to just covering up toxic portions of the antigens. So we've changed the structure of viruses. So now we've neutralized them. We've made them harmless. The third way is to stimulate local change. Inflammation. Inflammation helps spread the infection or helps to prevent the spread of infection. We're going to see IgE antibodies. We see mast cells, which are going to help with IgE antibodies. Mast cells have IgE antibodies on them. Remember that mast cells produce histamine. Remember, histamine is vasodilation. We also have inflammatory chemicals that cause edema and also vasodilation. Here is just kind of like a summary slide. We have the antigen-antibody complex. That reaction is going to trigger, agglutination, precipitation, or neutralization. We can see the complement activation system, phagocytosis, inflammation, cell lysis, and opsonization. Just another summary slide of what we talked about. So immunotherapy. The only thing that I'm going to cover on this slide, we're not going to go into immunotherapy. We've already talked about what cytokines are. We're not going to talk about, any of this. We're not going to talk about monoclonal antibodies, except for mono means one. So a monoclonal antibody is a single type of antibody. If you know anything about Crohn's, or if you've seen any TV commercials, there's a drug out there called Humira that will that's useful in Crohn's disease. Humira is basically a monoclonal antibody. And that's all I'm going to say about this slide. All right, so remember that we have a primary response and a secondary response. Remember that secondary response is those memory cells. So with our primary immune response, that's our first encounter with that antigen. Our plasma cells, our B cells, are going to release IgM and IgG. And then, we do that with our third line of defense, our specific lines, our specific immune system, so our humoral and our cell mediated, they're going to kick in. We're going to see our memory B cells be produced and stuff like that. But our plasma cells, it's going to take a while for our body to figure out what the exact type of virus or bacteria that we're dealing with. And so we'll see that when it's going to take some time for that virus to replicate and the bacteria to replicate and find its way to our cells and attach itself to the cell, creating this the antigen receptor linkage, and for all of that cytokines to be released, and the T cell activation and the B cell activation. So it'll take about 5 to 10 days for antibodies to appear in our bloodstream. And you'll actually see them in our bloodstream for several weeks. So they start to appear in about 5 to 10 days in our bloodstream, and you'll see that they peak about 10 to 12 days after exposure to a particular antigen. And just so you know, that's how vaccines work. Vaccines typically stimulate your primary immune response. But remember that we create these memory B cells, which are going to lead to our secondary immune response. So once we are exposed to that same antigen, our body has already done this whole immune response thing. And so we are going to see that now, instead of this 5 to 10 days, we're gonna start to produce these antibodies in 1 to 2 days because of these memory B cells and memory T cells that live in the lymph nodes. These antibodies, these memory cells can live for many years. And so it's because of the memory B cells and the memory T cells that you don't suffer from like chickenpox for the second time or third time again. So like the varicella zoster virus, that's the chickenpox virus. That's going to be why we don't actually get chickenpox again. So here you can see the primary immune response. In our bloodstream, we get exposed here at time 0, and then we'll be able to start to see, measure the antibodies at about day 5, but they'll peak somewhere, around here, day 12, somewhere. Excuse me. But with subsequent exposures, because of those B cells and T cells, you'll be able to measure those antibodies in our bloodstream much quicker. Here we're at day 2-ish, whereas here we were at day 5, day 6. And then we actually peaked a lot higher up here than we did with our primary immune response. All right, so we also have these things with immunity. we can break it up into how we acquire that immunity and whether or not that immunity is temporary or permanent. So if we and how we acquire it, is it natural or artificial? If we um, if we got the disease and we recover from the disease, that is natural immunity. Or if we got it from our mom, from breast milk or something like that, that is a naturally acquired process. Artificially acquired is from an injection, like gamma globulin or something like that, or a vaccine. So those are, more easier to understand than the act than whether or not it's permanent or temporary. So if you got a disease, you naturally acquired that disease, or if you got an injection or like a vaccine, that is artificially acquired. Now, we can classify it as active immunity or passive immunity. Active immunity is basically permanent immunity based on obtained via antigen contact. And it's evoked when and memory B cells are produced. So the body, we may or may not get sick, but our body has built up immunity to protect against those foreign agents. Now, passive immunity, it's temporary. And that's usually because we were exposed to the antibodies. We were not exposed to the antigen itself. And this is usually passed on from the mother to the fetus via breast milk. So we can have naturally acquired active, naturally acquired passive, artificially acquired active, and artificially acquired passive. So if I got a disease and I recovered and now I have permanent immunity, I would say that that is naturally acquired active. Naturally acquired passive, let's say that I got those antibodies from mom's breast milk, and they are going to provide short-term immunity. They don't stimulate an immune response. Artificially acquired active immunity. So that's typically vaccines. So we were given a vaccine that contained a weakened or a dead pathogen, and that's going to kick up our immune system without giving us any signs or symptoms of the disease. Now, artificially acquired passive, this is when we have injections of anti-serum containing antibodies or antitoxins. And this is very short term, and it does not stimulate the immune response. And one thing, here, and with COVID and everything, I'm sure you've heard of herd immunity, H-E-R-D, herd immunity. This is when we give vaccines given to the majority of the population to halt the spread of infection. So as long as the majority of the people are immunized, hopefully the disease will not progress. So we also have these things called hypersensitivity reactions. Basically, they are exaggerated immune responses to harmful or to non-harmful antigens. Typically, people are pre-sensitized to a certain antigen. We have 4 different types of hypersensitivities. So these would be like your allergic reactions to pollen or to peanuts or something like that. Typically, they can affect almost anyone, but certain people are, it's like when you say, I'm allergic to whatever. That's kind of like what we're talking about here. And we have four different types, four types of hypersensitivity reactions. Type 1, basically you're going to see this immediately. It's a type 1 immediate reaction, hypersensitivity. You're going to see an increase in your IgE response. It is a specific reaction. It occurs, you're going to see this happen when you have within minutes of contact. Your mast cells are going to dump out a ton of histamine. Remember, mast cells, histamine, vasodilation, huge drop in blood pressure. You're going to feel some signs and symptoms. You potentially life-threatening, anaphylactic shock. These are the people that need EpiPens. So some other symptoms. You might see hives, hay fever, asthma, eczema, gastric disturbances. You see the hives on the face and the neck. Asthma, your lungs are going to start to close up. Type 2 hypersensitivity. This is an antibody-dependent cytotoxic reaction. So you're going to see IgM or IgG. So your transfusion reactions are a type 2 reaction. This is where your antigen binds to a specific cell that's going to cause phagocytosis, and that complement system is going to lyse that antigen. A type 3 hypersensitivity. It's an immune complex reaction. Again, you're going to see IgG and IgM. This is where you're going to have that antigen antibody complex form in different tissues. That damaged tissue, we're also going to see phagocytosis and that complement binding. But that antibody complex can't be cleared from the body. You'll see that in like rheumatoid arthritis, glomerulonephritis due to streptococcal infection, systemic lupus erythematosus, SLE, or lupus, type 4 hypersensitivity, delayed reactions. These are usually going to involve your T cells. They can affect anyone. So this is where you have repeated exposure to a specific allergen like contact dermatitis, multiple sclerosis, type 1 diabetes. It usually takes about 48 hours to occur. You have repeated exposure to the same allergen that's going to cause this type 4 reaction. And basically that allergen is going to activate those T cells or those T lymphocytes in the skin. That's going to cause these chemical reactions that cause eruptions and inflammation of the skin. Just a summary slide of the 4 different types of reactions. Here is picture form of an immediate reaction allergy. This is going to be a type 1 reaction. Mast cells, remember mast cells release histamine. All right, transplants, tissues and rejection. So transplantation, we do transplant a lot of body organs, different things. Bone, corneas, kidney, liver, pancreas, heart, bone marrow, skin, lungs, just to name a few, uterus. And afterwards, typically you're going to be on immunosuppressive drugs to prevent an antigen reaction reaction. When we look at donation, we have to match you up with the donor versus the recipient. And if you're well matched, you might not need to be on immunosuppressive drugs for life. If the bone marrow stem cells from the donor are given along with a transplant. And we'll talk a little bit about we'll talk about it now. I'll just move this down. I'll just move this down here. All right, so when we talk about matching you up, we're actually talking about these MHC antigens. Remember we had the MHC1 and MHC2 when we talked about T cells? So that's what we're talking about. How do those match up? So the greater that antigenic difference, the stronger and faster your rejection is going to be. So you want the donor and the recipient to pretty much, be very, very compatible, to have a low antigenic difference. When we do transplants or grafts, if you have like an open heart surgery, if you do like an open heart surgery and you have due to like a coronary artery bypass graft, they're going to take a vessel from your leg, most likely, and use that as your new coronary artery or whatever vessel you're getting replaced in your heart. Because they're taking it from yourself, that is called an autograft. Now, if you have an identical twin, and an autograft is going to be the best place, because if it's coming from you and going to you, there's not going to be an MHC antigen difference. So you're not going to have an antigenic difference. Now, if you have an identical twin, that's going to be the next best thing, because your identical twin has the same DNA as you. So you're not going to have an MHC mismatch or an antigenic difference. So when we have that graft, if your identical twin donates something to you, that is called an isograft. And because your DNA from you to you, or from your identical twin to you, those would not cause any type of rejection reactions. Now, if you have a donated organ from someone else other than your identical twin, that would be called an allograft. And if you have, we do transplants from animals other than humans, that would be called a xenograft. And both allografts and xenografts could lead to a rejection reaction because they don't share our DNA. And so that's why we'd have to look at that MHC compatibility, and hopefully that antigenic difference is low enough. And that's why we need to be on immunosuppressive drugs, because that donor tissue might be recognized as foreign. And if we have that tissue rejection reaction, that recipient's immune system is going to start to go haywire, and it's going to try and destroy that foreign tissue. And it's going to look like how that cellular immune response against antigens, that T cell response, is going to start ramping up. And we have this thing called graft versus host disease. And that's just damage to the recipient's tissues from a transplanted tissue, such as bone marrow. So when we have that transplant reaction, because of that antigenic difference, it's called graft versus host disease. Here's just a little summary. An autograft is from yourself, an isograft is from an identical twin, an allograft is another human, not yourself, and a xenograft is from a different species. Autoimmunity, remember, that's an attack by the immune system against its own tissues, Remember, those B cells and T cells get vetted by the red bone marrow, and somehow one of those non-vetted B cells, T cells escape that red bone marrow. And those cytotoxic T cells attack the body's tissues, organs. We have different autoimmune disorders that attack different types of cells. A lot of different theories have been talked about, but we don't know much about autoimmune disorders. We've barely scratched the surface. Here you can see a bunch of different autoimmune disorders. One thing that people don't know is that type 1 diabetes is an autoimmune disorder. The beta cells of the pancreas are destroyed by your own body. So with here for my exam, for the exam purposes, you're going to have to know the disorders. I'm not going to ask you about symptoms or the antibodies against, but you're going to have to know the disorders only. So with HIV, human immunodeficiency virus, basically it's a virus that's going to break down your immune function. It's a silent disease. You could have HIV for years before you start even noticing any signs or symptoms. And left untreated, it's going to progress to AIDS, which is acquired immune deficiency syndrome. And then as your immune system starts to break down, you're going to see these things called opportunistic infections. Opportunistic infections are basically things that your body should be able to fight off that they can't anymore because you have no immune system. So what happens in HIV is that the virus itself is going to attack macrophages. Remember, macrophages are just phagocytes that kind of clean up cellular debris and all the dead stuff, and viruses and stuff. So the virus actually attacks the macrophages. And then it's also going to attack helper T cells. And then as your T cells begin to decline, your B cells can't produce antibodies due to the lack of cytokine activation. Remember, your helper T cells produce these cytokines or those colony stimulating factors to help B cells to help B cells proliferate. Now, if you don't have the B cells anymore, your B cells can't produce antibodies. So now your B cells are ineffective in the humoral response, and now your T cells are ineffective in your cellular immune response. So basically now HIV kind of kills off both your humoral and your cellular immune responses. They are now inefficient or ineffective. And then HIV also will start to kill off your cytotoxic T cells. And then you actually don't die from the virus. You die from, if you die, nowadays you live until, you're 90, 100, or you get hit by a bus or something like that. You don't die from HIV. You don't die from AIDS anymore. Typically, we have medications that help. But if you die, you're going to die from these opportunistic infections that are a result of your deficient immune system. And I think by now we know modes of transmission of HIV: sexual contact, sharing needles. You can actually get HIV from milk from infected mom, or if mom has HIV and delivers baby. If the blood is contaminated or if the tissue has HIV and you receive that as a donor. I would say that this last one is going to be, with modern technology, we're not going to see this anymore. We don't receive, the blood bank's good about screening blood for HIV. All right. Lifespan changes. Your immune system is going to kind of decline early on, just because, remember, your thymus gland is going to shrink, and also as you get older, your thymus gland changes from glandular tissue to adipose or connective tissue. So your immune response is going to decrease as you get older, which is going to cause you a higher risk of infections. So your T cell numbers decrease, your activity levels decline. Remember, your B cell numbers don't necessarily change, but your helper T cells are responsible for your B cell numbers as well. So your antibody response becomes a little bit slower. IgA and IgG increase, but your IgM and your IgE decrease. You might need an extra dose of vaccines. You might have more autoantibodies being produced. Therefore, you might develop autoimmune disorders. All right, and that is it for the lymphatic system. If you need anything, please let me know. There's two of my dogs. Mickey is on the left and Lucas is on the right. The other dog is Indy. She's not pictured. We got Indy. This was last Christmas. We got Indy after Mickey and Lucas, or after this picture was taken. So, all right. Let me know if you need anything, and I will see you guys later. Bye.